The following program is part of a college course. These telecourses allow you to earn college credits at home. For more information, contact a college or university near you.
Engaging, dynamic, dramatic. We see life on every blade of grass and in every face of the crowd. But in this diversity lies another world unseen. The hidden world of cells. All living organisms must react to a constantly changing environment. This holds true whether it's our survival or a championship game on the line.
I can remember in college we played Clemson University at Clemson, and there was at least 80,000 people in the stands. It was really loud, and there's a lot of distractions going on, and it's hot, and people are tired out there.
How does an athlete perform in hostile conditions? How do any of us survive in a challenging world? The answer lies in the smallest units of life.
Our body is composed of various organs, and those organs are composed of different kinds of tissues. And the tissues themselves have as their fundamental component cells.
Everything an organism does can be explained by the actions of its cells. We move, breathe, think. In fact, we exist due to the synchronized efforts of our body's cells.
Cells come in a variety of different functions and different types. And if you're really going to understand the function of the human body, what it's doing and how it's doing it, then you have to have a really good understanding of the different kinds and varieties of cells that exist.
We find over 65 trillion cells in one human being alone, each with its own role to play. Delicate brain cells to transmit messages, disc-like blood cells that enrich the body with oxygen, elongated muscle cells that contract to move limbs.
If you were to come over here, we have some more examples.
The form of these cells often reveals their function, as we see in the specialized components that allow us to hear.
When it comes to cells of the ear, it's really an incredible machine. How is it that we are able to hear certain sounds in the context of a very noisy environment?
It was loud, and at that time I was lined up correct, but I wasn't anticipating that play. And I heard my coach yell, "Watch the curl flat." And I was aware to hear him and focused on the play, but I had zoned out the crowd.
The crowd is shouting, they're roaring, because they want that receiver, of course, to catch the ball and then go for a touchdown. Well, you have to be able to block out the crowd, and you have to be able to listen to possibly your teammates, and you have to be able to listen to anyone else who may not be your teammate who's coming up from behind. Well, how do you do that?
To answer questions such as this, scientists use electron microscopes to analyze the structure of specialized ear cells.
What we know is that there are sense cells, which are very, very different from the supporting cells. The sense cells have these little hair bundles. Another group of cells that are very important in the inner ear are the nerve cells that actually make contact to the sense cells. And those two form what we call sensory neural hearing.
In the world of complex organisms, form reveals function. In the microscopic hairs of these sense cells, we find the receivers which collect sound waves. In the long, skinny reach of nerve cells, we find the circuitry which transmits impulses between the ear and the brain. This type of cell diversification is mirrored in much of the animal kingdom. For instance, the ear cells of a frog reveal a form quite similar to the ear cells of a human, or even those of a bird. In a similar way, the skin cells from a frog, a bird, and a human all look alike because they perform the same basic functions. Nature found a series of effective cellular designs and stuck with them. Yet even in this remarkable diversity, most cells share the same basic characteristics. Imagine we could enter one of the skin cells of this athlete here.
Welcome to the inner cell and the hazy world of the cytoplasm. Keep a sharp eye out for organelles, the functional components of the cells. There's the cell's powerhouse, a mitochondrion. Mitochondria transform sugar byproducts into ATP molecules, the main form of energy that cells can use. The membranous compartments floating about come from this Golgi body. Inside these layered sacs, the cell refines and packages proteins, fats, and other important materials. Where do these materials come from to begin with? Meet the endoplasmic reticulum. You're looking at the cell's main assembly line and distribution center. On its rough areas, these tiny dark bodies called ribosomes forge the proteins that are so crucial to life. The instructions for building those proteins come from perhaps the most important organelle of all, the nucleus. Its porous double membrane keeps the cell's DNA accessible but separate from the cytoplasm. DNA molecules distribute the unique molecular blueprints used to build each different protein. It's the ultimate in quality assurance, in one of the most efficient factories on Earth.
While we find the same organelles in each of our body's cells, their function may require more of some and less of others. For example, the constant activity of muscle cells requires more energy-producing mitochondria. The cells of our intestines are filled with pockets of digestive secretions. The intricate designs of each different cell determines what role it will play in the game of life.
Hearing my coach give me that read was the most important thing because I keyed out the whole crowd. And they ran the play, and I got an interception, and that put us in the lead. Well, I'll tell you something that's rewarding is after you focus out the crowd and you make a big play, you want to hear them screaming for you.
Just look at a cell and you see life, beating with its persistent duties, insisting that its daily processes continue like clockwork. What makes such a tiny trooper so tenacious? Well, packaging.
The membrane surrounding cells is absolutely critical to life. We have to hold what's inside the cell in and keep what's outside the cell out. And that membrane is what does it. Now here's a good example of drying out. We're almost ready to get rid of that.
Dr. Robert Heath and his colleagues study plant physiology, including how to grow plants from cell cultures and without soil. These experiments allow us to observe cells in a controlled environment.
Hydroponics is the ability to grow plants without soil. It's, as the name suggests, water culture. The roots extend down into water where you can change the nutrient level, you can add anything, and you can add various gases to the solution to change the root structure, rate of growth, and development.
Using hydroponics, scientists can produce a mature plant in just 7 days. But it requires the painstaking delivery of the right combination of nutrients. And in their microscopic journey to each cell, these nutrients must cross a formidable obstacle.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
So what we have to have in that membrane is our selective materials to allow a transport to occur. The cell's membrane keeps most things out, but allows what it needs to penetrate into the membrane so it can be utilized.
Dr. Heath's plants require a precise mix of inorganic nutrients, such as nitrogen, potassium, and calcium. Dissolved in water, we call these nutrients solutes, and nature has devised ingenious methods for chaperoning these solutes through the cell membrane.
Diffusion is a fundamental physical process. I think we're all aware that if somebody opens a perfume bottle in one side of the room, you can smell it in the other side after a time. Molecules move from a higher concentration to a lower concentration, naturally, through a molecular motion that occurs regardless of life, regardless of us.
Diffusion results from the random collision of molecules. In a natural search for more elbow room, they bump and shove each other from areas of high concentration to areas of lower concentration. Just as these collisions cause perfume or smoke to diffuse across a room, they can also push nutrients through any cell's membrane.
Smaller molecules can move directly through the membrane as they travel from a higher concentration to a lower one. But larger molecules and ions can't slip through the membrane. Lucky for us and life itself, the cell can transport these materials in other ways. For example, some proteins in a cell's membrane act as channels for specific ions or molecules. These channel proteins don't use energy at all. They simply allow the materials to naturally diffuse from the side with more solutes to the side with less. Whether the direction is out or in depends on where the concentration is higher for each different solute.
The normal pathway is from outside to in. That's the passive diffusion, passive transport method. The cell requires no energy. The energy is obtained from the excess concentration on the outside.
As diffusion brings solutes further inside the plant, their concentrations build. But the inner cells may still require more nutrients. However, with diffusion now working against them, the cells must fight to get what they need, transporting substances from a lower concentration to a higher one.
In this case, the cell uses transport proteins which need a boost from an energy molecule. This actually changes the shape of the proteins, causing them to pump different molecules and ions across the membrane, now from the side with fewer solutes to the side with more.
The cell is faced with higher concentrations inside than outside, but it still wishes more nutrient. It gets that nutrient by expending energy to pump, to pull in that excess. That is really the difference. Is there energy expended to pull in material?
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Just as cells move nutrients in, they can also move waste materials out. For example, a plant cell produces far more oxygen molecules than it needs. Thus, plants break down water and release oxygen into the atmosphere.
The study of plant physiology is absolutely critical to us because life on Earth is dependent on plants. Without animals, life would go on. Without plants, life would very rapidly cease. If we don't understand how plants work, how they produce the foodstuffs, the gases, detoxify the air, then we won't understand how to maintain life under a variety of conditions.
All life depends on the extraordinary actions of the cell membrane. Just as plant cells produce a cross-current of nutrients and waste, our body cells import and export the compounds we need to survive. In this way, the membrane becomes more than a shield. It's a gatekeeper, opening and closing its portals in response to the cell's needs. Whether we find that cell in a larger body or in a sea of the smallest organisms on Earth. Over 3.5 billion years ago, Earth's primordial oceans teemed with primitive life, bacteria. These were simple cells, much smaller and without the complexity of those which make up our bodies. Yet these tiny organisms still thrive in unseen multitudes all around us, even in the ground we walk on.
Well, in an area the size of a football field, say down to about the first only 4 inches, one would expect to find about a billion bacteria. Microorganisms interested me because of their tremendous diversity.
Microbiologist Dennis Vogt studies the life we find beneath our feet, including bacteria. These cells, which scientists call prokaryotes, have coexisted with humans since we first walked the earth.
There are many interesting stories about how prokaryotes have affected people. We could probably look at historical events such as Napoleon's defeat at Waterloo. It was supposedly due to a salmonella infection, that his troops were not up to their best fighting form. The bubonic plague that devastated Europe in the 14th century was a result of a bacterial infection.
In ordinary soil, we find diverse bacteria, both menacing and benign. For example, Clostridium botulinum produces a toxin that is deadly to humans. In its purified form, just 8 ounces could wipe out the entire human population. On the other hand, scientists use Streptomyces, another group of bacteria, as the basis for many life-saving antibiotics. Bacteria display an amazing diversity, yet their prokaryotic cells reveal a structure fundamentally different than the eukaryotic cells of more complex organisms, such as an amoeba, an orchid, or an owl.
Generally speaking, prokaryotes are quite a bit smaller than eukaryotes. For example, a bacterium is roughly the size of a mitochondrion.
This human cheek cell towers over the tiny bacterial cells around it. Though there are many variations, about 1,000 average-sized bacteria can fit into a typical eukaryotic cell. Yet despite their size, bacteria efficiently accomplish all the same functions of life that more complex eukaryotic cells do. They obtain and use energy. They reproduce. They respond to changing conditions. All this in a very simple package.
The general feature of a prokaryotic cell that makes it different than a eukaryotic cell is its lack of any type of organelles.
Prokaryotes are hardly more than lipid-bound sacs of cytoplasmic soup surrounding strands of DNA. On the other hand, eukaryotes require discrete membranous and non-membranous parts called organelles to organize and separate chemical processes. In fact, eukaryotes are the only cells with membrane-wrapped organelles, such as mitochondria for harvesting energy, lysosomes for digestion, and a nucleus for DNA isolation and protection. Without organelles, bacteria can reproduce faster, which allows them more opportunities to adapt and survive in harsh conditions.
They are known for surviving in some of the most extreme environments. For example, many of the deep thermal vents in the ocean are excellent habitats for sulfur oxidizing bacteria and methane producing bacteria. Many of them actually grow at boiling temperatures. Bacteria have been found in salt pools, generally where eukaryotes are not found because of the extreme salinity, and they've been found in environments that are extremely acid. In the battle for survival, prokaryotes are favored over eukaryotes because of their much simpler structure.
Prokaryotes lack another eukaryotic trademark, the cytoskeleton. The cytoskeleton gives these complex eukaryotic cells their shape and internal structure, just as the human skeleton supports our bodies. Composed of different protein bundles, this interior scaffolding extends from the nucleus to the plasma membrane. Primarily in animal cells, the structure of the cytoskeleton depends on microtubules, which anchor organelles and play a key role in cell division. These microtubules also give rise to the flagella and cilia, which organisms use to move about and stir up surrounding food. Because prokaryotes lack a cytoskeleton, their much simpler flagella are anchored in the cell's membrane. Still, regardless of their simplicity, these tiny bacteria play a critical role in the cycle of life.
The role of prokaryotes in the soil might be thought of as being at the absolute bottom of the food chain. They're the ones that are most important. They're the ones that take molecular nitrogen from the atmosphere and reduce it to forms that can be used by plants and then eventually by animals. They're the ones that are the start of the global nitrogen cycle. They're also the ones that do the same in terms of sulfur.
And perhaps most importantly, photosynthetic bacteria produce much of the oxygen in the air, shared by almost all living organisms. From football players to frogs, from budding plants to bacteria, we're all composed of the same things: cells. Whether elegantly simple or surprisingly complex, these cells go about the business of life. In their varied forms, we see the building blocks that assemble the rich diversity of nature. In their microscopic machinery, we find the processes that produce and cycle the necessities for survival. All life rests on these delicate parcels of nature, too small to see.